Okay, so good afternoon, everybody. And this will conclude our classes on microbial metabolism. This is where we ended up last time. And this is a summary of all of the processes that we discussed, which end up producing 32 molecules of ATP from one molecule of glucose. We saw all of our pathways: glycolysis, intermediate step, then the citric acid cycle, Krebs cycle. The point of all of these was, first of all, to produce a small amount of ATP, but most importantly of all, to produce masses of reducing power. That is, hydrogen electrons carried by electron carriers, which were then donated to an electron transport chain in the process of aerobic respiration. Oxygen is the terminal electron acceptor. And that process produced molecules of ATP in addition to the few that had been produced by substrate phosphorylation. And what we ended up with was a sum total of about 32 molecules of ATP. So I want to tell you that there are a few complications to the story, naturally, because this is biology. Everything gets complicated. And the first little complication to mention is this: that there are certain organisms which, under anaerobic conditions, and conditions where there is no oxygen, can continue to use aerobic processes in order to produce ATP. They still use the electron transport chain, etc., to produce ATP. And the way in which they do it is they still use oxygen. It's just that they don't use free oxygen. They don't use oxygen dissolved into the cytoplasm. Instead, they abstract oxygen from a compound which has oxygen as part of it. So, for example, they very often utilize nitrate, or they can utilize nitrite even. And what are they doing? They're taking away an oxygen from there to use as a terminal electron acceptor, or they're taking away an oxygen from here to use as a terminal electron acceptor. They may also there are some which will also use oxidized sulfur compounds, and they will reduce them by taking an oxygen away from them, and that acts as the terminal electron acceptor. This yields less energy than aerobic respiration, and but it's still worthwhile doing if they can do it, because it still produces a substantial amount of ATP. And we refer the enzyme here is nitrate reductase. The enzyme here is nitrite reductase. And you may recognize those names from lab, because these form the basis of very important discriminatory tests. We test to see whether organisms have nitrate reductase or nitrite reductase. And this acts as a discriminatory test in the lab. The whole process is this special type of respiration, and it's called anaerobic respiration, which is a bit of a tautology, but it's because they don't use free oxygen. They use oxygen which is already bound to another compound. Now, we need to distinguish that very clearly from the processes of fermentation. So first of all, in the complete absence of oxygen, and if an organism is unable to produce one of the enzymes to allow anaerobic respiration, many organisms are still able to produce a small amount of ATP from the process of glycolysis. You'll remember that our net gain from glycolysis was 2 molecules of ATP. And that is enough for many organisms to proceed with their other metabolic processes. Organisms which operate under these circumstances are referred to as being anaerobic organisms. And there are some organisms which are obligate anaerobes. They cannot be in the presence of oxygen. They don't have the mechanisms to utilize oxygen. as a terminal electron acceptor. There are others which can temporarily use this process when they put into anaerobic conditions and then return to aerobic respiration when oxygen is present. And those we refer to as the facultative anaerobes. So that's fine. These organisms, they can utilize glycolysis as a source of ATP, but there's a major problem with doing this, because you remember that there were two products of glycolysis. The first was ATP, which is very desirable. That's fine. The other is NADH. And in order to produce NADH, you have to have a source of NAD plus as a substrate. And if you produce, as you produce NADH, especially if the NADH is not being sent off to an aerobic respiratory electron transport chain, you accumulate NADH, but you deplete NAD plus. And NAD plus is vital in many, many metabolic reactions and pathways. So under the conditions of where they are simply using glycolysis, organisms use up all of their NAD plus. And this is a very dangerous situation, because all sorts of metabolic pathways grind to a halt when they don't have a source of NAD plus. And the organisms then have to do something about that. They have to do something to regenerate NAD plus from NADH. And the way they do it is the process of fermentation. So this is important for you to realize, that there is often people confuse the use of glycolysis and fermentation. They think that one and the same thing. They are not. Fermentation is an entirely separate process to glycolysis, and its entire point is to regenerate NAD plus for the cell to use in its other metabolic pathways. So our end product of glycolysis was 2 molecules of pyruvic acid. And the end products of glycolysis now can be used to produce NAD plus if we take NADH here and we use that hydrogen to reduce the pyruvic acid, or something that has been derived from pyruvic acid. It varies a little bit depending on organism. And what we're going to end up with is an end product. This is the process of fermentation. And we're going to end up with an end product. And most importantly, we're going to end up with NAD plus to fund all the other metabolic pathways. So this leads to a situation which is important for us as microbiologists in many, many ways, also as human beings in many, many ways. So here is our typical situation. Here we have the normal processes of glycolysis. Here we're using up our NAD plus and producing NADH, a little tiny bit of ATP. Now organisms have to push this pyruvic acid through fermentation pathways. And there are many of these pathways. There are many ways in which organisms actually do this. We'll just consider the most important. Whatever they are, whichever pathway they use, you're going to see that we're going to oxidize NADH, and we're going to produce NAD plus. And we're going to use all of the hydrogens that are derived from that to produce another compound. And it's going to be a reduced form of pyruvic acid. And it depends very much on which organism you're talking about. The first group of fermentations are called the lactic acid fermentations. And the simplest of all are those which just produce lactic acid. So those that only produce lactic acid, just one kind, are called homolactic, the homolactic fermenters. This is homolactic fermentation. But there are others, many, which produce lactic acid plus a whole slew of other compounds, which we'll see in a minute. And those are called the heterolactic organisms. So the other type of fermentation, which of course is hugely important to human beings, is are those which can produce alcohol. And this is alcohol fermentation. And this produces ethanol and it produces CO2. Those are both byproducts. Here it's shown here. But don't worry about all of the intermediates and everything. Just have a look at the end products and what we end up with: ethanol there, carbon dioxide there, and we use the NADH to reduce this compound here to ethanol. So again, what we're doing in the whole pathway, taking NADH, regenerating NAD+, reducing, oxidizing NADH, producing NAD+. It just depends on the organism as to which product you end up with. Either lactic acid in the lactic acid fermenters or alcohol in the alcohol fermenters, with a little bit of a twist, okay, because some of them can do both, as we'll see right now. These are very useful reactions. First of all, they're useful in the laboratory because we can use them to distinguish one organism from another, and they are characteristic of certain organisms. Second of all, of course, we utilize these very much, especially in food and in food preservation. So the first is that these that produce lactic acids, especially the homolactic fermenters, we utilize in things like sauerkraut, all of the fermented foodstuffs, kimchi, any of these foodstuffs. The lactic acid creates an acidic environment which prevents pathogens and spoilage bacteria from growing in food. It also lends that acidic taste, which human beings actually find quite attractive, just like sauerkraut, right? In agriculture, it's hugely important, and it has been ever since mankind settled down and became agrarian. The reason for that is, for instance, in the Northern Hemisphere, we face a very long winter when food, grass and hay and everything are not available to cows and horses and everything that are being kept. And what farmers will do is during the summer, when there's an abundance of hay and grass and everything, they will lay it down, allow it to ferment. And what happens is they encourage the production of lactic acid producers, homolactic bacteria especially. And that is sauerkraut for horses and cows and everything, and can be kept over winter and fed to them. And it is extremely useful because they can actually also use the lactic acid as a nutrient. So the heterolactic, those that produce a variety of acids, for us are very useful as well because they are used industrially to produce some of the strange compounds. Alcohol fermentation, we use it in two ways. Now, this is not necessarily just bacteria, because of course we mostly utilize yeasts to perform alcohol fermentation, but it's exactly the same process. And the first is that they produce alcohol. In all of the alcohols are produced this way. But second of all, they produce carbon dioxide. In drinks, that is the source of the fizz. In beer and that sort of thing, is the carbon dioxide produced as a result of fermentation. But we use the same process, for example, in baking, because that is how we get bread to rise. Bread is a rich nutrient source from flours and everything else. Bread, the flour is processed so that it produces gluten, which is like a glue. The bread dough is actually like a gel. And trapped in it are the yeasts, which are going to ferment. They're going to ferment, they're going to produce alcohol, for sure, but they're also going to produce carbon dioxide. They'll produce bubbles of carbon dioxide, but this is a gel-like substance, the bread dough, traps the gas, and the bread rises, but then we put it into the oven. And what happens is that all of that gas expands so that the bread rises completely and cooks. And at the same time, all of the alcohols are cooked off. And that lovely smell of fresh bread baking, that a substantial amount of that smell is actually the alcohols cooking off of the bread. So these are very, very useful processes for us. And I'll show you that some of the compounds that are produced during the process of fermentation, first of all, they are characteristic of particular bacteria. But second of all, some of them are extremely useful to us, and these organisms are cultivated industrially specifically to produce these. Well, think about the first lot that are really easy. The homolactic organisms are things like lactobacillus. Lactobacillus is the one that we use in many of our fermentation processes, like cheese production, sauerkraut, all of those things. We use lactobacillus. And that word, that part of the word there, lacto, tells you that what they are doing is they're using lactose, which is milk sugar. And they end up producing lactic acid as a result. And Streptococcus, lactobacillus, bacillus, these are typical homolactic fermenters. Yeasts are alcohol fermenters, and they only produce ethanol. They produce ethanol and carbon dioxide. But there are other organisms which we're going to see produce a mixture of all these different compounds, all sorts of different compounds. This one, Propionibacterium, produces an organic acid called propionic acid. It also produces acetic acid or vinegar and carbon dioxide, and can also produce free hydrogen as part of this process. Clostridium, which we're going to hear a lot about when we talk about bacteria and health, the Clostridia are obligate anaerobes. They are obliged to use glycolysis and fermentation. They cannot grow in the presence of oxygen. They will not, in fact, survive in the presence of oxygen. And depending on the Clostridium you're talking about, they produce a variety of compounds. Anything that produces a mixture of acids is called a mixed acid fermenter. So these are mixed acid fermenters. And the Clostridia may be mixed acid fermenters as well. They produce a variety of very useful industrial compounds. Butyric acid smells like old socks. Butanol, acetone, isopropyl alcohol, all of these things, as well as CO2. So these would be mixed acid fermenters. Common lab bacteria, the gut bacteria such as Escherichia and Salmonella, and some others as well, produce a variety of compounds, and they can produce a mixture both of alcohol and mixed acids, as well as carbon dioxide and hydrogen, free hydrogen. This one here, Enterobacter, also a gut bacteria, and it produces ethanol and lactic acid, formic acid, and it produces a substance here called butanediol and acetoin. And I have mentioned these because you may in lab do tests which discriminate this group here from this group here by looking for that. And these are the methyl red Vosges-Proskauer tests. So keep an eye open for those when you do them in lab. This is what you're looking for. You're looking to see whether they produce just mixed acids, like these here, or whether they produce mixed acid plus these compounds here. Okay, so that's the question of fermentation. Make sure you've got that clear in your heads, okay? So now we have discussed how ATP is produced by substrate phosphorylation. We saw that through all of those reactions in glycolysis and then in the Krebs cycle. Those reactions were all substrate level phosphorylations, and there's some others in the cell as well. We saw how abundant ATP is produced from aerobic respiration. Those are the processes associated with those electron transport chains in the membrane, which utilize oxygen as the terminal electron acceptor. We mentioned briefly the anaerobic respiration, which is kind of a special situation. And then here comes our third major way of producing ATP in biology. This is limited to those organisms which are able to utilize light as a source of energy. In other words, these are processes which are tied to the process of photosynthesis. This is an autotrophic production of ATP from the process of photosynthesis. And here I want to run through the process of photosynthesis very briefly, because you are actually going to do a take-home exercise, which I'll elaborate on a little bit in a moment. So I'm just briefly going to remind you what the process of photosynthesis actually entails. Remembering that photosynthesis, first of all, evolved in prokaryotes. And second of all, there is a very large group of extremely important prokaryotes called the cyanobacteria, which utilize photosynthesis. They produce a huge amount of the oxygen which is present in our atmosphere. And of particular importance are cyanobacteria, which are planktonic, which are in the oceans, in surface waters of the oceans. So let's remind ourselves about the whole process of photosynthesis. First of all, it is a membrane-bound process. And the reason for that is that we need to have our proteins and everything, all the substances needed are embedded or associated with the membrane. Again, so that what can happen is we can have differences on one side of the membrane compared to the other. And so here's briefly the structure of the systems embedded into the membrane. We have two units which contain the chlorophyll molecules, which are called the photosystems. And here's photosystem 1, here's photosystem 2. The numbering is only because that was the order in which they were discovered. So photosystem 1 was discovered first, photosystem 2 was discovered last, but photosystem 2 is actually the first one in the chain. So I hate to use the word miraculous, because nothing in nature is miraculous. It is there for a purpose, and it evolved for specific reasons. And can be explained. But the truly marvelous aspect of photosynthesis is not all this stuff. It's this process right here. Because what photosynthetic organisms can do is they can take water and they can actually split it. Now, if we want to split water into oxygen and hydrogen, we have to put in a huge amount of energy to do so. Just immense. It is an immense and very difficult process. If it was as easy as it is in plants, then we would have abundant hydrogen to use for energy. But in fact, we have never learned to actually do this as easily as plants do it. What they do is they take hydrogen from the water. What are they using the hydrogen for? They're going to snatch electrons from the hydrogen, and they're going to leave protons. They're going to accumulate protons. And this is how they do it. They take the electrons from the hydrogen and they feed them into the system. Now, what chlorophyll consists of is a molecule with some very loose electrons associated with it. And that is the nature of a pigment. A pigment is any substance which interacts with light. And the way chlorophyll interacts with light is that some of its hydrogen electrons get so excited by the light waves that they actually fly off the chlorophyll and can be used to reduce the next substance here. And this substance is able to reduce, it is able to oxidize the chlorophyll. And then it passes, the electrons pass through a series of chains. And what we are doing is we're actually, at the same time, accumulating protons here. We're accumulating protons, which are not present on the other side. In addition, there are some proton pumps here, which are able to pump protons into the space here. So now what is familiar to you? This is similar to aerobic respiration. It's the same kind of idea. We're accumulating protons on one side of the membrane, which are not present on the other. And as these electrons flow through these systems, again, they lose energy, and protons can be pumped to this side here. In the end, the energy from this first electron loses so much energy that we need to renew the electron transport chain. And what happens is, here is the photosystem 1, actually the second one in the line, but it's photosystem 1. And photosystem 1 behaves in much the same way, except that this chlorophyll molecule here loses electrons, and the electrons are actually utilized in another system, in another short electron transport chain, which ends up producing this substance here, NADPH. What is NADPH? But an electron transport. It's a transporter. It's reducing power. It's actually a very powerful amount of reducing power. So what have we done? Well, look at what has happened. Here at this chlorophyll here, we've lost an electron. It's gone into this whole system here to produce NADPH. You can't have that and have a free electron, which has come from here running along. So this electron that ran down this chain here now replaces that electron there in the chlorophyll. So this system here, in addition to being acting as a way to produce NADPH, can also be used to produce ATP, because what we've done is we've accumulated lots of protons here, and we have exactly the same enzyme, ATP synthase, embedded into the membrane here. We allow those protons to flow through to equalize on both sides, and we harvest the energy from those protons as they pass through the ATP synthase, and we produce ATP. Not very much. Okay, this is not very much. Now, I want to add, we've put all of this together into one system. These can actually be separated from one another by mechanisms we don't need to talk about. And both of these systems can be utilized just to produce ATP if the plant cell really needs it. So the end product of photosynthesis, a little bit of ATP. That is true. But much, much, much more importantly is this NADPH, the reducing power. And all of this stuff is going to be sent off now to a system which is going to be able to manufacture organic molecules. This is our energy stage. This is how the plant accumulates energy and converts light energy into chemical energy. And we're now going to follow this as it goes to the next stage of photosynthesis. This is called the Calvin cycle, named obviously for the person who first described it. But the Calvin cycle is really the most important part, I suppose, of the biological processes in terms of the ecology of plants and everything that eats them, because this is this part of the process which is actually going to produce organic molecules from carbon dioxide and water. Where did we utilize water? We utilized water here. We utilized it in order to actually replace the electrons which have streamed through here. And we've utilized the hydrogen electrons there to replace a chlorophyll electron, which we kicked out to move through here, to replace the electron which we kicked out here. The byproduct of that is oxygen. So that is why plants produce oxygen. But the oxygen is not utilized by the plant. It is released as a byproduct. And instead, all of those products, the reducing power NADPH and the ATP, are now used in this very, very metabolically expensive process. What happens is we're going to take carbon dioxide, a simple molecule, and we're going to push it through this cycle, and we're going to push for every single carbon dioxide molecule, we're going to run the cycle 6 times. How many carbons does carbon dioxide have? It has 1. If I run it 6 times, theoretically, I can end up with a 6-carbon compound. What is the first 6-carbon compound that springs to mind? Glucose. That is the point of this whole cycle. We're going to end up producing a molecule of glucose. And in order to do so, we're going to have to utilize 6 molecules of carbon dioxide and a huge load of energy. But it doesn't matter. It doesn't matter how much energy it takes. Why? Because the energy is free. It's free because the photosynthetic organism is able to gather light, sunlight, and convert it from light energy to chemical energy. So I'm not going to go into this in enormous detail. I'm just going to point out to you that, first of all, it is a cycle. That means it has to regenerate its first substrate. And that first substrate is called ribulose bisphosphate. And take a minute to write that down. Ribulose bisphosphate. And look at it, how many carbons does it have? 1, 2, 3, 4, 5. If I now move this ribulose bisphosphate and I unite it with a carbon dioxide, I end up with a 6-carbon compound. But that 6-carbon compound is extremely unstable. And it breaks down into this molecule here, which is 3-phosphoglycerate. And that is our first product of the Calvin cycle. We are now going to run this through here, and we never mind all of the machinations. We're going to end up, every time we run this cycle 6 times, we're going to spit out 1 molecule of glucose. There's only a couple of things on this diagram that I want you to remember. First of all, we're adding glucose, we're adding carbon dioxide to our initial reactant, which is ribulose bisphosphate. And we're utilizing, to do that, what has been called the most important enzyme on the whole planet, which is ribulose bisphosphate carboxylase oxidase. They haven't even put the oxidase there, but its proper name is ribulose bisphosphate carboxylase oxidase, commonly referred to as Rubisco. And it is the responsibility of Rubisco to unite ribulose bisphosphate with a carbon dioxide. That is done 6 times because we start off, actually, our first product is that 6 carbon breaking down into 3 into a 3 carbon, which is then passed through this whole cycle 6 times in order to produce 1 molecule of glucose. But look what happens. We're running it 6 times, right? We have to add 6 molecules of ATP, 6 molecules of NADPH. Then this here is called the regeneration phase, because what we need to do is regenerate the ribulose bisphosphate. Another 3 molecules of ribulose bisphosphate of ATP to produce our ribulose bisphosphate again. It's a very expensive metabolic process, but it doesn't matter because our energy source is abundant and it is free. So is the small molecule that is needed to do it, carbon dioxide. It's free and abundant. So this is the process which ends up producing our organic molecule, glucose, from simple precursors. All the plant needs, it needs sunlight, it needs carbon dioxide, and it needs water to do it. So I want to say one other thing about plants, or things that when I say plant, I mean anything that uses plant photosynthesis. That's a cyanobacteria and everything as well. Don't forget that at the same time as photosynthesis is marching merrily along and producing glucose, the autotrophic cell still has to respire. And the autotrophic cell still has to respire in order to produce abundant ATP to utilize in all of its other metabolic processes. Photosynthesis during the day runs in parallel with aerobic respiration in the autotrophs. At nighttime, when there is no light, only the respiratory pathways are running. Then the photosynthetic pathways start up again as soon as it is light. Okay, so that's only a brief overview because I want you have done this in biology before, the whole process of photosynthesis. But now I want you to work through this all on your own. And you are pleased to go, I hope this works. You are pleased to go to this site here. And if you have never been here before, this is a really marvelous site. And this is the Howard Hughes Institute. And they have a massive online presence, all sorts of things about biology, microbiology, virology, anything, all sorts of things. You can, when you're done with this exercise, then you can go and have a look and see if there's other stuff that interests you. There is a worksheet to go along with the exercise that I want you to do. And you will see that it takes you through logically through the whole process.
Almost all life on Earth is solar powered. Plants, algae, and some bacteria.
And it'll take you through the chemical processes, leaf structure of chloroplast.
Let's take a closer look at a chloroplast.
Lighter reaction.
The thylakoid.
Carbon reaction, etc.
The Calvin cycle takes place in the chloroplast.
So now I need to get back. How do I get back? I don't know. Let's try this. Oh, okay, oh yes, there we are. Just give me a minute bumbling around. Okay, so here, so this is an interactive exercise. There is a worksheet which I will put up online for you, and you will have 2 weeks in order to do this. Okay, and then I want you to hand it in. This is for extra credit, but it is also homework. So you must do this, and it's for 1.5% of your extra credit. Not only that, this is examinable, and there will be questions about photosynthesis. Even though I skipped over it myself, you are to derive as much as you possibly can from working it through, and you should have a basis for this from biology. If you still have your biology, access to your biology textbooks, and you need to use those, that's fine, but there is full explanation about photosynthesis in your in your microbiology textbook as well. Okay, so 2 weeks, please get that back to me. All right, so I hate to tell you this, but yes, there are some other complications about aerobic respiration. One of them being that there are some alternative pathways which run either instead of or in parallel to glycolysis. And the reason for this is a little bit complicated. Some of these pathways, in fact, we think were the original pathways, which then evolved to glycolysis. But quite apart from that, these pathways, the best known of which are the pentose phosphate pathway and the entner-Duderoff pathway. These are extremely useful to the cell because they produce many precursors of for other metabolic pathways. And they're especially important in synthetic pathways, as I'll show you in a minute. I'm not expecting you to remember much detail, but both of these two pathways, the pentose phosphate pathway and the entner-Duderoff, feed to anabolic pathways and very often create monomers of things like the proteins and things like that. So some organisms have both. Some organisms possess both pathways, glycolysis, pentose phosphate, and entner-Duderoff. Others possess only one or the other, either pentose phosphate or entner-Duderoff. In your lab, you won't do any discriminatory tests to see, but these can be used to classify organisms as to whether they possess these or not. I'm only going to gloss over them. I just want you to know that they are there. One important thing about them, and one thing that makes us think that they're relatively primitive pathways, they produce not NADH, but NADPH. NADPH is most commonly produced in the plants, but not exclusively in photosynthetic organisms. I keep saying plants, and I mean to say photosynthetic organisms. The NADPH is mostly commonly produced in things which are photosynthesizing, but it does occur in heterotrophs as well. And this is an example of where it actually occurs. The pentose phosphate can operate simultaneously with glycolysis if an organism has it. And they produce a lot of intermediates, especially for the production of nucleotides and amino acids. The entner-Duderoff pathway is rather similar to glycolysis. This is one of the pathways we think may be like a fossil metabolic pathway in a way. And it does produce, just as glycolysis does, it produces ATP and ultimately pyruvic acid, but it also produces NADPH as the electron carrier. And it's not tied to glycolysis at all. It can operate independently, and is characteristic of these genera of bacteria and can be used as a discriminatory test in Pseudomonas rhizobium and Agrobacterium. So now don't worry about the detail of what I'm going to show you now. I just want to point out to you that these intermediates of glycolysis and of the pentose phosphate pathway and of the entner-Duderoff are used in many, many, many different metabolic pathways. And this is one of the ways in which pathways are actually tied together in the cell. From the glycolysis itself, it is some of these intermediates can be moved out to form, for example, glycogen. This is an anabolic process, so it requires an input of energy in the form of ATP. And glycogen, remember, is a very, very large molecule. and it's synthesized by joining glucose molecules together. And in order to do it, this is used glucose 6-phosphate, which is an intermediate of glycolysis. The fructose 6-phosphate, which is one of the intermediates I asked you to remember the name of, is utilized to produce peptidoglycan. Peptidoglycan is also a polysaccharide, and you'll remember it's a component of the cell wall. Lipids are very easily manufactured from intermediates of glycolysis and of the Krebs cycle. And the most important unit here is this here, acetyl coenzyme A, which is used to manufacture fatty acids. And these are put together with glycerol, which is an alcohol sugar, which can be produced from intermediates of glycolysis. And those are joined together to form lipids. Amino acids are also manufactured from these precursors, but very importantly, amino acids are produced from the pentose phosphate pathway and from entaner-deuteroff. This may, in fact, be one of their most important functions in the cell, in those cells that have them. But amino acids can also be produced from the intermediates of the Krebs cycle as well. Never mind about all of the detail, just note that pyrimidine and purine nucleotides, which we will hear more about when we talk about DNA structure, nucleic acid structure, etc. These can also be synthesized from intermediates of glycolysis, the Krebs cycle, and from pentose phosphate pathway or the entaner-deuteroff pathway. So there are many sources of substrate to produce these. Okay, so having said that, I just want to finish off by telling you a little bit about how metabolically diverse the prokaryotes actually are. Now, I've said it over and over and over again, that there is not a single metabolic process that didn't evolve in the prokaryotes. So we can expect that as we look through the prokaryotes, we can see that they are extremely adaptable to many, many different environments, utilizing many different sources, both of carbon and of energy. And we've already talked a bit about it because we've seen, first of all, we've talked a lot about heterotrophic bacteria that utilize a fuel like glucose or whatever as a source of hydrogen and of reducing part. But they also are utilizing the carbon to build their biomolecules. So those are the heterotrophs. But we've also talked about the autotrophic prokaryotes, such as the cyanobacteria, which utilize carbon dioxide as a source of carbon and the sun's energy as their source of energy. So it actually gets a little bit more complicated. Everything I tell you can always be elaborated on, all right? We start with the simple and then we move through the more complicated. So let's have a look at all living organisms. And we can divide all living organisms into groups depending on what they use as their energy source. So think about ourselves. Where do we get our energy from? We get our energy from already created biomolecules. Those are chemical compounds, things like glucose, etc. They are chemical compounds. That is where we derive our energy from. And because we derive our energy from a chemical, we are referred to as chemotrophs. On the other hand, other organisms utilize light as a source of energy, and these are the phototrophs. They're utilizing light as a source of energy. So here's our two groups. They each have their source of energy. But now where do they get their carbon from? Carbon is essential because it is the backbone of all of our biomolecules. So it's essential that we have a source of carbon. Where do these two groups get their carbon from? Well, that can be very complicated. Because look at us, think of our own physiology. We derive our carbon from already created organic compounds. And because of that, we are called chemoheterotrophs. That is us. We are chemoheterotroph. And we are a chemoheterotroph because we use already created organic compounds as a source of carbon. And then we use them as well for a source of energy. So that's us. But having said that, we have to ask another question. And that is, how do they use it? How do they utilize these already created organic compounds as a source of energy? Because there are various strategies. And the first thing we notice is whether or not they utilize oxygen as a terminal electron acceptor or not. Well, we know that most eukaryotes like us are utilizing aerobic respiration. So we are utilizing oxygen as the terminal electron acceptor. And that is by far the commonest situation because it's a very, very efficient way of operating, of producing your ATP. So that's all animals for a start. Almost all fungi is the same, and many, many bacteria and protozoa. They all utilize this system, but not all. There are some which do not use oxygen as the terminal electron acceptor. Instead, they use an organic compound as the terminal electron acceptor. And those are the fermentative organisms, for example. They're utilizing an organic compound and they're oxidizing it. Those are the processes of fermentation that we've already seen. And those are the fermentative bacteria, like all of the fermentative things like streptococcus and all sorts of things like that. However, and that's relatively easy to understand, but there are some which utilize an inorganic compound, and they are going to reduce an inorganic compound such as sulfur or something like that as the terminal electron acceptor. And that's this group here. They utilize an inorganic compound. So even in this group, in the chemoheterotrophs, we see that there is a substantial diversity. And all of this diversity ends up, where does their what is their terminal electron acceptor? They are all utilizing some form of electron transport chain in order to produce their energy. They biologically used for the energy. Okay, that's the heterotrophs. They were all those organisms which use organic compounds. But there are other chemotrophs which use carbon dioxide as a source of carbon. And these are called the chemoautotrophs. Now, I'll simply explain it like this. These are organisms which are able to perform true autotrophy. True autotrophy is defined as utilizing carbon dioxide as a carbon source in order to produce an organic compound. However, these are a little bit different, to things that utilize photosynthesis, because they're not utilizing light as a source of energy. Instead, they are utilizing other chemical compounds as a source of energy to do this. They're going to unite carbon dioxide with water, and they are going to produce an organic compound. They are autotrophic, but they're going to use a chemical compound as a source of energy. And what they do is they oxidize. The simplest one to think about is this one here, and that is hydrogen sulfide. Well, look at hydrogen sulfide. What is it? It's H2S, we can utilize the hydrogen from the H2S instead of utilizing water. We're going to use the organism is going to utilize that as a source of reducing power, and is going to produce organic compounds. CO2 plus H2S is going to release sulfur, but in the process, it's going to be able to build up these carbon atoms into an organic compound. That's a simple example. There are many, many variations on this theme in the prokaryotes. There are many chemoautotrophic prokaryotes, and their characteristic is that they utilize carbon dioxide as their carbon source, but they utilize chemicals as their source of energy. Okay, so those were our chemotrophs there. Now, the other group are those which use light as a source of energy. And these are the phototrophs. And there are two groups of phototrophs. The first, although we divide them on, again, on their carbon source. What do they use as their source of carbon? There's one group, which we won't spend much time talking about, but know that they are there, which strangely enough are called the photoheterotrophs. Well, if they're heterotrophic, they're utilizing an already created carbon compound as a source of carbon. They're utilizing a biomolecule as a source of carbon. So they are heterotrophic, but they don't use that compound as a source of energy. They utilize light as a source of energy. And there are these strange groups here, the green non-sulfur and purple non-sulfur bacteria. And we are not going to say much about them, just know that they exist. Again, what we're saying all of the ones that we're talking about now, they are using light as a source of energy. And there are those which now utilize carbon dioxide as a source of carbon. And those are the ones which are much more familiar to us. These are the photoautotrophs. They're utilizing carbon dioxide, light energy to create organic compounds. But there's a little bit of a variation as to where they get their reducing power from. Where do they get their hydrogens from? Those that utilize water as a source of hydrogen there are called the oxygenic photosynthesizers. And that's the photosynthesis that is familiar to us. It evolved in the cyanobacteria, and it produces oxygen as a byproduct, because what they do is they take away these hydrogens there from water and use them, they bind them to the carbons that they're grabbing from carbon dioxide to produce their organic compounds. So that should be familiar to us, and be even more familiar to you once you do your photosynthesis exercise. But have a look here and you'll see that there are organisms which do not use water as a source of reducing power. And these do not produce oxygen as a byproduct, but they many of them, for example, use H2S as a source of reducing power, as a source of hydrogen. And they will lay down sulfur. They're called the anoxygenic photosynthetic bacteria because they do not produce oxygen, but they may accumulate things like sulfur. There are some other compounds that they can use as well. I know it's confusing. So what I want you to do, this is a summary of it. You can use this little table to remember exactly what each of them are. First of all, our very, very familiar groups, the photoautotrophs, which use light and carbon dioxide light as a source of energy and carbon dioxide as a source of carbon. Those are familiar to us, the oxygenic ones, cyanobacteria, the plants, algae, all of those sorts of things. But then remember that there is a group which does not use water as a source of reducing power, and those are the anoxygenic organisms. Just know that they exist, okay? and what is their characteristic. There's another group, the photoheterotrophs, still use light as a source of energy, but they depend on organic compounds for their carbon. And again, remember that they exist. The photoheterotrophs break down organic compounds for their carbon, but they utilize light as a source of energy. Chemoautotrophs are those which use some inorganic chemical like H2S, ammonia, there are all sorts of other compounds which can be utilized as a source of energy. And that is done by the chemoautotrophs. And they still utilize, they're autotrophic, they still utilize carbon dioxide as their source of carbon. It's just that their reducing power comes from somewhere else. And by the way, in the chemoautotrophs, it's not always hydrogen. They can still grab electrons from other things, but it's most commonly hydrogen that they use. A good example of, for example, are the iron oxidizing bacteria, but there are thousands of chemoautotrophs. The chemoheterotrophs are those which use an already created organic molecule as a source of energy, but also as a source of carbon. And that's us. That's how our cells function. And fermented bacteria, which have been fermenting, that would be them as well. But most commonly, it is the things which are familiar to us. Okay. So remember, please, that you have that exercise to do, and that with that, I finish off because somebody is banging on my front door.